So, we are going to talk today. I thought in lieu of sort of starting something new just before the exam, I thought we'd sort of take a lecture and kind of wrap up a little bit of what we know about the sensory processing from a different perspective. So the last, I don't know, 7 lectures, we've been talking about sensory systems, all the different kinds of sensory systems, a lot of content, sort of knowledge, and now we're going to talk a little bit about, in sort of a more general perspective, when we say something, the visual system is coding for visual information, what do we mean by that? We kind of throw around the word coding a lot. Let's talk a little bit about what it means to code for something. And I'm not, we're not going to get into a very technical discussion. You could put a bunch of, information scientists and computer scientists together to talk about, what exactly information means. We're not going to get quite that technical, partly because I don't think it's all that productive. But just to kind of get you thinking about the idea that those of us who are sort of sensory physiologists, perform experiments, we ask questions, we want to know how neurons respond to stimuli. But oftentimes, it's not always easy to know when you have an answer. It's not like, a geneticist wants to know, what a particular gene codes for. So they go and clone it, and they can come up with an answer. You can get a very concrete answer. The Human Genome Project can come up with a very concrete answer. There are subtleties because there's variation and stuff, but you kind of know when you have an answer. And in neuroscience, the answer kind of depends on the eye of the beholder. Like, there are many levels at which you could answer the question of, what is area X doing? And in one sense, that's kind of fun and fascinating, and in another sense, it's kind of frustrating because you never really know when you have the answer. But anyway, so it's kind of an interesting thing. So we're going to talk about neural coding, what exactly neural coding is. And so usually when I say neural coding, I mean what kind of information is being encoded in the activity of a particular neuron. And we're going to look at this mostly at the single neuron level. But of course, single neurons don't encode a visual scene. A collection of neurons encode a visual scene. And we've talked a little bit about hierarchies, how processing occurs at different levels. Okay, well, we're going to kind of start with single neurons. And so the idea is that a neuron actually acts like a little computer, right? We talked a little bit about synaptic integration, how it receives multiple inputs, and then combines them in some way, and then produces an output. And this is really sort of what a computation is, right? Okay, so we're going to talk about this in terms of the idea of encoding a stimulus, because we've just talked about sensory stimuli, but you can equally apply this to what we're going to learn next, which is the motor system, where you have to produce some type of motor output. Okay, but the whole idea is that you have some sort of input stimulus that you have to encode in some way, and then individual neurons or groups of neurons will have a response. Okay, so this sort of stimulus into response is really what most neuroscientists kind of mean when they say what's being encoded here. Okay, we do that by measuring those responses. And we'll talk a little bit about how measuring those responses, we can get back to what was the stimulus, which is kind of what the nervous system is doing, right? You don't have, in a lot of ways, you don't have any a priori knowledge about the environment. What you know about the environment are the signals that are coming in, that's being filtered through your sensory system, and then you have to make sense of it. Okay, so in a way, the brain is kind of sitting here, or at the next level, trying to make sense of all of the information that's coming in through your sensory system. And finally, the end goal, really, is to create a behavioral output. As sensory physiologists, oftentimes we kind of think, okay, well, once I know what the encoding is, I'm kind of done. But really what the animal needs to do is it needs to move in the environment. It needs to trap its prey. It needs to move out of the way. Or it needs to get away from a noxious stimulus. So in the end, what you're looking for is a behavioral output. Okay, and that occurs through sort of a long chain of sensory information processing. sort of take an example of a very simple computer. I'll sort of make an argument that the brain is a computer in some sense. Okay, and in what sense do we mean? So a very simple example is that of a thermostat. Okay, so you have some temperature sensor, so that's your incoming sensory information. And then the thermostat processes that information in some way. And the basic processing is really quite simple. If the temperature is low, your behavioral output is going to be turning on the heat. If the temperature is high, the behavioral output is going to be turning on the air conditioning or turning off the heat. Okay, so that's kind of an idea of a very simple system. And in a couple of cases, we've talked about reflexes that essentially are this system, right? A lot of sort of homeostatic mechanisms in physiology are basically this system. Okay, but when we talk about code, we talk a little bit at a little bit more abstract level of what do we mean by a code. So here's an example of a code. Okay, Morse code is a code that you take some sort of stimulus, which are the letters, and you create some sort of representation of that stimulus that you can use to communicate. Okay? And so our basic idea is to take the dots and dashes, which are our neural activity, and reinterpret that as what was the original stimulus. Okay. And so there's a number of different ways that this can be done. The brain probably uses every strategy it possibly can to perform these sorts of calculations. But we're going to talk about a few basic ways. We've talked a lot about rate code, whether you realize it or not. We've talked a lot about the rate code. And that's just the number of spikes, right? So we say larger the stimulus, the more spikes. And that's what we mean by a rate code. What's the firing rate, and how does it relate to the stimulus? And we'll sort of touch a little bit on what do we mean by information in this system. And then I'm going to kind of introduce you to the idea of the temporal code. And we touched on this a little bit in the olfactory lecture. And then we'll just go from single neurons to talking a little bit about populations of neurons. Right, so every nucleus essentially is a population. And then each of those talk to each other in a hierarchy. And then we'll talk a little bit about receptive field estimation. We talked about this a little bit in vision, and we'll sort of expand on that a little bit. Okay, so our sort of simplest case scenario, back when we talked about somatosensation and thermoreception, is the rate code. Okay, and I think you all probably have a really good feeling for what this is. That if you have, in this case, like a cooling response, so cold receptors, we talked about thermoreceptors. So cooling comes on and goes off. And the degree of cooling is reflected in the number of spikes, right? So here's your spontaneous firing rate. Remember, these are raster plots, so this is time on the bottom. And then each of these tick marks is meant to represent a single actual potential at that time. Okay, so here's your spontaneous activity. And then as you start to apply a cooling stimulus, you see an increase in firing rate. And I think you can all see that there are more spikes up here than up there. Okay, so firing rate represents, in this case, the amount of cooling. Okay. And the dynamic range over which you respond can really differ across different neurons. And we saw an example for this. So firing rate also depends on what range of stimuli we're looking at. So this was an example. I think you recognize this. This was the thermal sensation. So this was your standard thermoreceptors that starts to respond at relatively low stimulus and increases in the response. And so if we take the numbers of spikes here and make a plot, so this is response rate or firing rate versus temperature, so that's the amplitude of the stimulus, you get a curve that essentially looks like this sort of blue curve. Okay, and two things we can see. One is that there's a fairly limited range over which the firing rate changes. And above a certain level, we saw that the firing rate actually flattens out. So even though the temperature is increasing, the firing rate is not increasing. And so the range over which a neuron is responsive is known as a dynamic range. So that tells you what range of stimuli that neuron is encoding. Okay. In contrast, we've talked about the nociceptor, the thermoreceptor that's a nociceptor, that only responds at relatively high stimuli. So it doesn't respond very much, and then it starts to respond with more spikes, and we could make this plot here, right? So the dynamic range over which neuron 2 is responsive doesn't actually overlap with the dynamic range that neuron 1 is responsive. And so they have different dynamic ranges. And so that's essentially what we mean by one's sort of a standard thermoreceptor and the other's a nociceptor, right? Their dynamic range is quite different. And so neuron 1 will respond over this range, and anything below that, there's no response. Okay, and so you've kind of split up the stimulus space. So there's this concept of a stimulus space, which is the range of all possible stimuli in an environment. And you divided it up into dangerous heat, which neuron 1 is responsible coding for, and non-threatening changes in heat, which is what neuron 2 is responsible. Okay, so these are average responses, and I think everybody kind of has a good feel for that. What we haven't really talked about yet, though, is noise in the responses. Okay, so any biological system has a certain amount of noise in the system. It just doesn't respond exactly the same way every time. Either because the stimulus is just a little bit different, or this, the position, the activity level that you started with, that neuron had just fired, maybe it's going to fire a little bit differently. Anyway, there's noise in the system. Okay, and so if you apply a stimulus to the same neuron several times, each of these, trial 1, 2, and 3, these are 3 consecutive trials, it's going to respond with elevated firing rate if it's in its dynamic range, but it might respond a little bit differently. So the exact position or number of these spikes is going to vary. Okay, and in this case, they don't vary very much for this particular neuron, but say in another neuron that happens to be a noisier neuron, you're going to get relatively large variations. So here's a few spikes on this trial, lots of spikes on trial 2, and some middle number of spikes in trial 1. Okay, so this neuron is noisier than this one for whatever reason. Okay, so if you plot this in terms of its response rate, the noisy neuron, they could have the same mean response, so this is the same basic curve, but notice that the standard deviation bars are much larger in this case, right? So if the stimulus was here at 45 degrees Celsius, this neuron will respond with anywhere from about 10 hertz up to 40 hertz, somewhere in between. Okay, which one do you think is more, let's say, reliable? Which one would you rather use to encode temperature if you really wanted to know what the temperature was? The left one? Yeah, the left one, right? Sort of obvious. Because, why? Why would you want to use the left one? The right one has exactly the same basic shape.
Because the standard deviation is small.
Because the standard deviation is small. And what does that mean? So you're the experimenter, and you're sitting there, and you're taking measurements, and you take a measurement. Yes?
Is it more precise?
It's more precise, right? Yes?
It's going to fire the same for each individual temperature, while the second one won't be. It could fire the same thing for 2 different temperatures because the error bars are so large.
Right. So the answer was that, the answer that was just given was that if you have the same firing rate, it could have responded that way to 2 different temperatures, I think. Did I paraphrase that properly? Which is, I think, what the answer is more precise implies. Okay. Did you have something else? Somebody else had their hand up. Yes?
Could you encode information in the variation itself?
The question is, could you encode information in the variation itself? How would you do that?
So based on so if you have a system of neurons that are firing, right? And one of them serves maybe some type of coincidence detector. And so its spike patterns might have some type of mean average, but due to the variety of stimuli coming in or leaving the system, it might be more noisy than another neuron adjacent to it, which is more unilateral in its response. So my question would then be, is in a neuron that integrates that amount of information, which we could have more variable output, could it be encoding some type of information in its variability itself?
Let's see. Yes. So the short answer, so the question was, can you use the variability itself to encode information? And maybe as we go along, we'll point out points that you could do that. In fact, there's kind of a very popular theme right now in neuroscience that is looking at what they call correlated noise. And so the idea is that the variation that you see is caused by something, and that something might actually give you more information about the stimulus itself. But yeah, so the variation itself might tell you something about the stimulus, okay, which is sort of a little meta for what we're going to talk about today. But yeah, so basically, if you took a measurement, you know, say we measured 25 spikes, okay, you'd have a pretty good feeling for what range of stimuli cause that response if there's relatively low noise. Okay. So the basic idea, so this concept of using the neural response and inferring what the stimulus was is decoding. So we just talked about encoding, and so going in the other direction is decoding. So stimulus to response is termed encoding. Response, going back to stimulus, inferring the stimulus from the response is known as decoding. Okay, so if we, you know, measure, you know, in this case 35 spikes, we know that it had to fall somewhere on this range of the curve. And given the standard deviations, we know it has to be somewhere in this range. Okay, this little bit of the curve here, which corresponds to this little bit of the stimulus space. So we've gained information about this system. Okay, if we went back to, and so one way of looking at this is to say, out of the entire stimulus space, okay, we can basically eliminate the green component, and our uncertainty is limited to this little component between the arrows here. Okay, so we've gained all of this information about the stimulus. Okay. If we had a lot of noisiness, large standard deviations, okay, we observe the same spike rate, so we take one sample, observe a 35 hertz spike rate. Okay, we are really uncertain. We don't know. It could be the stimulus could be way up here on the curve or way down here on the curve. All of this falls within those error bars. And so we have a much broader range of potential temperatures that could have elicited a 35 hertz response. And so we've gained some information, right? But not as much as in the low noise case. Okay, so noisier responses have less information. Trial-to-trial noise gives you less information. Okay, but the amount of information that we've gained really depends on where you are in this curve, right? So we talked about a 35 hertz stimulus, which is on the very steep part of this curve. If we had made a measurement at, say, 2 hertz or 0 hertz, okay, we're down on this flat part of the curve. Okay, and that means that the temperature could have been anything sort of below where our threshold was. So we have a much broader range of potential temperatures given that response. And so the amount of information that you get from the response, the measured response, depends on where you fall on this curve. Okay? And the higher the slope is here, the more information you have. Okay, so the slope, where you fall, and the noisiness all determine what kind of information you can get. Okay, so we have this sort of general idea that we can measure information based on the stimulus curve. That if we're on the steep part of the curve and there's relatively low noise, the steep part of the curve means that 2 different measurements have a big difference in their responses. So small changes in the temperature means a big change in the response rate, right? Whereas at this very shallow part of the curve, The same difference in temperature gives you a very small difference in the firing rate. So the steepness of that slope really matters. All right. So far we've been talking about firing rate sort of abstractly. And so the term firing rate really kind of depends on what time window you're talking about. And different people will use different time windows, depends on how rapidly your stimulus is changing. So there's really kind of an experimental choice that you make when you're talking about firing rate. What time window are we talking about when we're talking about firing rate? Okay, so we talked a little bit earlier about the this is peristimulus or post-stimulus time histogram, PSTH. Okay, and that's simply taking all of these individual trials and binning or summing up the number of spikes per unit time. Okay, so here's, multiple bins. Okay, and so this is a post-stimulus time histogram. Okay, so here's another example of a number of rasters. There's like 30-something trials, and here's the post-stimulus time histogram. Okay, and one of the reasons that you would do this rather than just sort of generate a single number is that we know that firing rates are not a stationary thing, right? So you have a stimulus. Sometimes you can just set a big window, but we know that firing rates really change over the time course of the stimulus, or right around the stimulus time. And we know that partly we talked about so think of this as the time varying of the response, variation in time of the response. And that's particularly important when we talk about adaptation, right? So you can get a big response at the very beginning, which then tapers off, even if the stimulus is continuing. All right, so you could vary your parameters, and it could really affect sort of how you are visualizing the firing rate. So here's an example. This is sort of a classic way, which is where you just have square bins, and within each of these bins, you count the number of spikes over a large number of trials, usually. Okay, another way to do it is to take a little small window and do counting. So one of the problems with this method is that you've got very sharp divisions between those that may or may not have anything to do with the time variation in the stimulus. You could get very artificial variations there. For example, is this bin really much bigger than the firing at the next time? It's hard to know exactly which of those steps are important, which are sort of an artifact of the binning. Okay, and so one way to get around that is to use a sliding window, which is where you just take a little time window, and you count the number of spikes, and then you move the time window over a small amount that's smaller than the time window itself. So say you have a 5 millisecond time window, you move it over a millisecond and count again. You move it over a millisecond and count again. And that way you can kind of smooth over some of those rough transitions that might just be an artifact of the histogram. And so you could take the same data and you'd get sort of a smooth histogram like this. If you smooth a lot, you can start to lose some of the variation that occurs. So here, in this smoothed count, you have a very clear peak here, and then a trough, and then 2 peaks. But those second 2 peaks are kind of smoothed together in this case. So this is just doing the same thing, but using a much broader window. Okay. On the other hand, maybe this is a useful thing. So it really depends on what it is that you want to get out of the system. So here's an example from a visual response. So this again is a raster to a sinusoidally modulated visual stimulus. And these are 3 examples of how you could smooth the response. So the red is the least smooth, and you get a lot of this sort of jaggedy stuff. The green is somewhat smooth, and it's starting to look a lot like the stimulus. Okay, and then the blue, it just gives you basically the average firing rate over that entire stimulus period. Okay, so which one of these do you think represents the stimulus best? Red, green, or blue? Green. Yeah, most people would say green. Okay, so the choice of smoothing or windowing that you use in order to look at firing rate can have this tremendous impact on how you're looking at the responses. And so a lot of sort of very classical neurophysiology work was done using very broad bins, counting lots of spikes over a fairly broad time period. And as things have gone on and it's gotten easier to do a lot of analysis, you can do much more detailed analysis. That starts, I think, people really started to look much more at the timing components. Okay, so one example, we talked about this in the olfaction lecture. We talked about the temporal patterning. So here's sort of the same system, slightly different representation. So here in green, these are olfactory neurons. This must be the antennal lobe, so that olfactory bulb equivalent. And the green are all responses to odorant 1. But here we have 4 neurons responding to different concentrations of odor 1. And you can, I really don't, I don't want to go into this too much detail, but you can really see that the timing of the spiking here differs across different neurons and differs according to the concentration. So in this sense, you may have sort of the same average firing rate, but the differences in the timing of those spikes tells you about the odorant. I think maybe the most striking case is, I don't know which one. This one's pretty striking. So you have sort of a mild inhibition here, inhibition followed by excitation, and then a deep inhibition followed by heavy excitation, and so on. And then if you compare these responses across 2 different odorants, you can really detect which odorant was it based on the pattern of the responses. Okay, and so they've done a lot of information theory on this, but looking at these patterns really gives you much more information about the sensory stimulus. And so this is an example of a type of temporal code, where we take into account when exactly the neuron is firing. Okay, this is another example of a temporal code. So this is with auditory stimuli. So the idea is that you can give a fairly low rate of clicks, each of these lines. This is the auditory stimulus. This is a click at a relatively low repetition rate. This is a click at a relatively high repetition rate. And there are some neurons, these are cortical neurons, that are very good at responding at a precise time after each click. Okay, so you can derive the frequency of those clicks directly from the auditory response. And as the clicks increase in repetition rate, it continues, especially at the beginning, but maybe you kind of lose it at the end. Okay, and so this is an example of a synchronized neuron. And so over a fairly broad range of click rates, you can derive the click rate directly from the cortical response. This is in contrast to another neuron, which is using the same stimuli, is not responding to every individual click. You have an increase in firing rate. So in this case, it's rate coding the rate of temporal clicks, but it's not actually encoding the clicks themselves in a temporal code. That makes sense? Okay, so this would be kind of a good example of a rate code versus a temporal code. And this sort of distinction between rate and temporal coding is a little bit artificial, because any temporal code can become a rate code depending on what type of window you're looking at. So you can look at each of these, and the time scale really depends on what you're talking about. So every sort of smooth post-stimulus time histograms, if you use a very narrow window, you're essentially talking about a temporal code. Okay. So how can we tell the difference between whether we have a temporal code or a rate code? Okay. So one way that we can do it is we can go through the idea, well, what if we temporally jittered all of the spikes? Okay. So if you can temporally jitter the spikes, and it doesn't make any difference to the information, then you don't have a temporal code. Okay. If it does make a difference, then you're gained information from the timing of those spikes. And so this is something that people frequently do. They look at whether you can rely only on the temporal information, or you really need, only on the rate information, or if you really need the temporal timing. Let me back up just a little bit here. So for example, in this example, here you have an increase in the firing rate, but jittering the timing of those spikes doesn't take away any information in the system. You get the same amount of information, no matter what the timing of those spikes are. Whereas in this case, if you jitter the timing of the spikes, you lose some information about repetition rate. You may still retain some information because it looks like there's more firing here than here if you averaged across the entire system. So in this case, it's not really a completely either or. In a few special examples, you can actually jitter the timing of the spikes without any changes in firing rate. So you can have one, you can have the other, you can have sort of a mixture of the two. Okay, so that's single spikes. So what about populations? This is an example that actually we'll come back to when we start doing motor responses. These are rasters recorded in motor cortex in response to hand motions of the animal, in this case a monkey moving a joystick. Okay. And this is sort of a classic example of population coding. So the idea is that if you record from a large enough set of cortical neurons, that you can determine what direction that the animal is moving the joystick. Okay, so let's start. So each of these rasters is, like 5 trials, okay? And the 0 is the onset of the movement. And because it's a motor system, the neural activity actually precedes the movement. So this is a premotor response. Okay. And these are all recordings from one neuron as the animal makes movements in each of these directions. Okay. And so you'll notice that if the animal moves the joystick to the left, there's a large response. If it moves it to the right, there's inhibition. And then as you go around the clock, the degree of excitation and inhibition changes. Okay. And so in this sense, we call the area in which you have the most excitation the preferred direction. Okay. So now what if we had a group of neurons that all had their own particular response pattern? Okay. This neuron has a preferred direction to the left, so that's this red arrow here. But imagine we had another neuron that had a preferred direction to the top, and another neuron that had a preferred direction to the right, and a fourth one that had a preferred direction downward. Okay, so if you recorded from all 4 of those neurons, actually let's start. If you recorded from one of those neurons, and you recorded a firing rate of, say 20 hertz, how sure are you which direction you were moving in? Right. Do you think you're moving probably to the left or the right? Left or right? Who says left? Who says right? Oh, come on, you gotta vote. Okay, let's say this is about 20 hertz. Okay, so if you measure 20 hertz, which direction is it moving, left or right? Left. Probably left. But you might have some uncertainty whether it's moving sort of up and to the left or down and to the left. I'm pretty sure it's left. Now, if you record from a second neuron, okay, maybe the up neuron. Okay, so it fires a lot when it moves up and very little when it moves down. Okay, and if you record from that neuron and you get a high firing rate, also as high as the leftward neuron was, which direction do you think you were moving? Up and to the left. Okay, so you just gained information about the movement just by recording from 2 cells. So you can imagine that if you have lots of cells, you can have a really good idea of what the direction of movement is. And this is the basis of the population code. That if you combine information across many cells, so in this case, the upward is firing 10 spikes, the leftward preference neuron is firing 10 spikes, the green one is firing 1, the bottom one is firing 1, you have a pretty good idea that this is the direction. And so if, and this was exactly what the experimenters in this system did, is they basically combined information in a vector code. So you've all had vectors before. So if you represent the firing rate as a vector with an amplitude and a direction, and you do a vector summation, you get a very reliable estimation of what the direction of the movement is. Right? And so if we have another example where we have high firing rate to the right, sort of slightly higher firing rates down below, you can get a vector response, a vector representation of that movement. And so this is what we mean by population code. Combine information across a whole group of cells. Okay, so this is one of the earliest demonstrations of a population code. All right. Okay, so receptive fields. Back in vision, remember, a few weeks ago, we talked about the receptive field of a ganglion cell. So this is an example of one of those receptive fields. An on-center ganglion cell. It's got an inhibitory surround. Okay, and we saw how you would determine that, right? So you shine little spots of light, change the amplitude of the light, and so on. And so that's one way to determine the receptive field. Okay, and that works pretty well for sort of early peripheral vision. And it doesn't work terribly well for higher order areas. And so in order to, especially for dynamic stimuli, so simple static stimuli are pretty straightforward, but once you start to get into more complex stimuli, especially if you start talking about natural scenes, right, and moving animals or moving scenes, things start to get a little more complex. Okay, so people have devised other ways of measuring tuning, measuring receptive fields in these cells. And so we'll just talk a little bit about how you would go about doing that. Okay, so far we've talked about sort of orientation tuning in the cortex, so you can build a little representative field. But what does this really tell you about a complex stimulus that looks like this, that may be moving across your retina? Okay. So to answer this question, people have turned to slightly more sophisticated methods known as receptive field estimation. Okay, and so the first thing that we can talk about is sort of a spatially static stimulus that turns on and turns off, right? And we've already kind of alluded to this before, that when you turn a stimulus on, you have an onset response, okay? This is sort of that sort of phasic response right here. But then as the stimulus maintains intensity, there's no really further responses here. So in one sense, it's adapted, it's phasic. So what's it really responding to here to? Is it responding to the presence of the light? I asked. No, really, what's it responding to? If you measured the response of this neuron at any given point in time, can you tell whether the light is on or off? Not really, no. If you're sticking your electrode in the cell and you notice a sudden of the response, what happened? There was a change, right? So the light came on. Since it's an on neuron, you can tell that the light came on. So knowing nothing about the stimulus, you can infer something about the world based on your measurement from that neuron. Okay, that's the decoding. Okay, and so importantly, this is responsive to the turning on of a stimulus. Or equivalently, the movement of a stimulus outside of its receptor field, into its receptor field, and then maybe out again. Okay, and so these neurons are really designed to detect changes in the light. Okay, and those changes we can represent in a temporal receptive field. So here's a spatial receptive field where on the retina is the light happening. And this is a temporal receptive field. This is what is the best change in the stimulus to elicit a response. Okay. So this is luminance versus time. And so the dashed line here is the onset of the response of firing a spike. Okay. And if this is the stimulus that precedes this point in time, you get a response. You fire a spike. That's what it means. Okay. And so you can also think of this as a filter. Okay. And so the way that this little cartoon is saying is that you can apply this filter to the stimulus, and when the filter matches the stimulus, you get a response. Okay. So it's like sliding this little filter along the stimulus. So when everything's off, there's no match between the filter. When you have that sudden onset, there's a pretty good match with the filter. The filter is looking for a rising phase. And then when the light stays on, again, it's a bad match to the filter. So it doesn't respond. Yes?
So are we supposed to be reading the temporal receptive field graph from right to left?
It's kind of backwards.
Oh, right.
Right? So this is the time of the activity. And you're trying to say, well, what stimulus preceding the activity is preferred?
So that's the big one is used stimulus sign and the small ones are the activity?
Oh, sorry, this is just a small version of this.
Okay, so that's
Relative to the stimulus. So here, if you apply the filter at this point of the stimulus, you'll get a big response. If you apply the filter at this point of the stimulus, there's a poor match, and so you get no response.
Is the filter like, what is the filter?
What is the filter? So the filter is kind of, it's basically a time representation, just like this is a spatial representation. It says at what point in time. So if this is, say, 1 millisecond from here to here, if there was a rise in the light stimulus at 1 millisecond, at a delay of 1 millisecond from that time point, you'll get a response. So this could be a longer delay or a shorter delay. It could have some flatness to it. The degree of negativity here means that if you have sort of, it gets dark and then light, that it responds even better. So the particular shape of this filter tells you something about the response properties. So for visual stimuli, this tends to be sort of relatively broad. For auditory stimuli, it's much, much more narrow. So auditory is faster than, yes?
So are you saying that like stimulus isn't static, that the stimulus they're introducing is varying too?
So the stimulus is the same in this example. So it's off, and then on, stays on, and then turns off.
Okay, so then how are they able to detect these changes in luminance then if the stimulus is the same? That's what we're down to.
How are we able to detect? You don't detect changes in luminance, in fact.
But isn't that what it's saying over there?
This is detecting, I'm sorry, you don't detect, you detect changes in luminance because you're looking for a rise in the luminance.
Yeah, but the stimulus isn't changing though. That's why I'm kind of confused.
The stimulus isn't changing. The stimulus is changing, but only at this point right here.
Oh, so that's why you're saying it detects the onset.
Right, right. So it's detecting the onset because that's where the change is. Yes.
Why is the second filter not at the
Oh.
after it turns on?
So you have to read this as we're going to take this filter and we're going to kind of slide it along.
Okay.
Okay. And at whatever point the filter matches the stimulus. So there's no match, no match, no match, match, fires, and then no match, no match, no match, and then it turns off.
Okay.
So how do we, let's move on from this. So this is, let's get into exactly how would you create a filter like that. So one really common way to do this is to do, in a very sort of intuitive way to do this, is to do what's called a spike-triggered average. So in essence, it's a spike-triggered stimulus average. So what's, on average, what's the stimulus that precedes activity? Okay, so let's walk through this. So yeah, so this note is like hand mapping. Hand mapping meaning you're Hubel and Weasel and you've got a little dot and you're moving the dot around on the screen. It's a pretty inefficient way of determining receptive field. Okay, so how else would you do that? And this also gets at this idea of kind of decoding. So what we're going to do is we're going to apply a stimulus. So in this case, this is a light stimulus that varies in stimulus intensity, so it sort of jitters up and down for short periods of time. Okay, so basically it's kind of like a flickering stimulus. All right, and you're going to record the neuron's response. So this little ball here is a neuron, and here's your electrode and your amplifier, and you're going to record spike times. Okay, so you record these spike times, you kind of do it over and over again. And then for every trial, you're going to go back and say, okay, for every spike that I had, what was the stimulus that preceded it? Okay, so the advantage of using a stimulus like this is you're just kind of, you're not biased in terms of what type of stimulus you're using. You're kind of using all of the stimuli. And you're jumping up and down, so you have all of the potential time changes in the response. Okay, so you're going to take every spike, and you're going to draw a little box around the stimulus, and you're going to say, okay, well this was the stimulus that occurred just before this spike. And you can do it again and again. So for every spike, you're going to draw a new little box, okay? And then you're going to average those boxes. And what you find is that on average, just prior to the spike, which is at time 0 here, just prior to the spike, on average, there was a dip and then a rise. Okay. And you can kind of ignore the backside of this curve, because once you've had a spike, it doesn't matter what the stimulus is. The really important part is that this dip and this rise. And so you can use this to make that type of a filter. And if you do that, that kind of response, and you do it over a space, a visual space, or we talked about auditory neurons using a frequency space, you can develop a real map of the responses. And this is really how people do visual stimuli response, receptive field estimation. Now, nobody does little dots. Yeah.
How noisy are these clocks on average too? Because I feel like.
You need a lot of data.
Okay.
You need a lot of data.
Okay.
Yeah. And the bigger the stimulus space, the more data you need. So you could need thousands of spikes. Once you have the data and you have the stimulus, it's actually fairly trivial to run this kind of an analysis. But you do need a lot of spikes, especially if you have a relatively low firing rate. It can take a long time to collect the data, and that's one of the drawbacks of doing it this way. Yes?
So for these filters, is a filter specific for a particular neuron or
Yes.
specific? Okay.
Is
So
a
it doesn't
filter
matter-
specific to a particular neuron? Yes.
So it doesn't matter what kind of stimulus you're given, so it's for this one neuron, this is always going to be the filter.
Yeah, so does it matter what kind of stimulus you're giving it? It matters in the sense that you want to basically cover the stimulus space. So if you only give a certain type of stimulus, then you're only going to get information about that area. So one of the keys to doing this kind of experiment is to use, oftentimes it's referred to as like a white noise stimulus. You have random variations of the spatial resolution can be very large, or the temporal resolution, you can have very fast transitions and sort of slower transitions. And so you really kind of want to cover the stimulus space in order to get the response for that neuron. That being said, there's often a lot of similarity in the sort of qualitative nature of a stimulus. So very often, you'll get something that looks very much like this, sort of very classical response. Sometimes it can be flipped if it's response, depending on what type of sensory stimulus we're looking at. You could have the filter go in the other direction so that a decrease in stimulus might elicit a response. So that can vary at all. Any more questions? All right, so now that we've talked about this, I want to kind of throw out some caveats about this type of work. Okay, so we've talked a lot about encoding a stimulus, but just because a neuron responds to a particular kind of stimulus doesn't necessarily mean that that is the neuron or set of neurons that are responsible for that type of encoding. So that's one thing to kind of watch out for. Okay, and then there's another concept called efficient coding, which is kind of related. And the idea is that, especially back in the 90s, people were very excited when they started doing these sorts of information theory. And they found, oh, isn't this interesting? This neuron is actually doing at its very theoretical limit for how well you could possibly do in encoding this stimulus. And so that was referred to as efficient coding. So on the one hand, that's very exciting because it means that over long evolutionary times, brains are really good at encoding certain types of stimuli. On the other hand, the sort of more biology, physicists were very excited about that. The more biology end of neuroscience said, well, brains aren't perfect. Evolution is kind of a messy process. And really, the animal doesn't need to be extremely efficient. It just needs to be good enough. You have to be good enough to get away from the predator. You may not have to determine where the predator is to a 0.001 degree arc, right? So there's sort of an argument for having a good enough strategy, even though some neurons are actually quite efficient. And then the third caution I want to kind of make is that, as an experimentalist, we really sort of love this idea of being able to take the neural responses and then infer something about the stimulus. And that's a really fun thing to do, and it tells you a lot about what information is contained in those responses. But that's not really what the animal is doing, right? So the animal, it doesn't, you've all sort of seen what's the word? The homunculus view, where you've got, the little man in your head looking at the activity in your V1 and seeing a little scene of the world. Lots of, popular science articles love to have that kind of view, and that's not really what you're doing. I mean, you kind of have that percept as a person sort of walking around the world, but that's not really what your brain is intended to do. It's intended to produce behavior. It's intended to, visually track something in your space. And so what the neurons at the next level are doing for the information at the level preceding it is not necessarily what we think of it as. It's not trying to decode that information, it's trying to use that information for further purposes. Okay. So, sort of from a more practical point of view, what does neural coding mean to, say, a patient? And so you could make an argument that, what we're kind of doing as neuroscientists is kind of esoteric. We want to know how the brain works. But this can have really important implications for things like neuroprosthetics. Okay, we're not really going to have a lot of time to talk about it, but I want to talk about it a little bit because this is something that I think is really sort of exploding in current neuroscience. So the idea is that understanding population decoding can be a very useful tool for things like neuroprosthetics. So this is an example of a woman who is quadriplegic. She has no use of her arms or her legs. And so she really can't get around. And so now people like this are receiving implants in their motor cortex. And you can decode the information, the neural activity from the motor cortex. And she's using this now to move a mechanical arm. So a prosthetic arm. So the signals are coming from an electrode implanted in her cortex, being processed, decoding, as we just really talked about, and that I just kind of dissed a little bit. So you're decoding that information in order to move a mechanical hand. And we'll talk about this a little bit later when we talk about motor patterns. But this is one example of, you know, how you really can use that kind of information for very practical purposes. Oh, and I just kind of threw this in because we were talking about maps earlier. Okay, and so the idea is that you don't really need a map for population coding. Right? You don't need sort of an orderly progression of activity across the motor map. So this type of decoding just doesn't rely on that kind of information. Okay, and then more broadly, concepts of coding and decoding is important for different types of neuroengineering, neuroaesthetic, brain-machine interface. We talked a little bit about cochlear implants, and I thought it'd be fun to kind of look a little bit at retinal implants. Okay, so now that we know a little bit about how these circuits work, it's interesting to say, well, can you take a person who has no vision and then stimulate in a way that the person can interpret those stimulations as a sensory input. Oh, and I just wanted to point out this Nicolelis TED Talk is actually quite good. I'm not going to talk about this too much, but I wanted to show you a little bit about retina. So I know you all have gone back and reviewed the lectures on the retina and horizontal cells and center surround and all that. Yeah? Oh, some of you have. Okay. So, so we talked about cochlear implants, but people are also working on retinal implants. And so I think switch to. And so this is a group at Carnegie Mellon that, and here they're using simulation to try and understand what the retinal implant, what would you see using a retinal implant? Okay, so this is basically an 8x8 matrix. And so here you're using sort of a normal visual scene on the left. And on the right are, the dots are activity at the sort of photoreceptors at that particular spot. And so just kind of notice what you can and can't get out of this activity. You kind of get an idea that there's an edge here, but you're not really seeing sort of these colored rings. Okay, and this is using a fairly low spatial resolution retinal implant. And it's black and white too. It's not, there's no sort of color information here. So you see the whiteboard, you sort of see, and you see the edges of the whiteboard, right? And when you move around, you sort of see the edges of where the door is. So any areas of very high contrast, you're getting a lot of responses. So now this is a 16x16 matrix, and now you're getting what? What do you see? What's different about this than you saw before? Yeah, it's higher resolution, which means you can resolve sort of smaller objects. You can see, you have more information about the edge there. You're also sort of getting a sense of visual flow, right? If you move around. Yes?
How are they getting this like processing into the brain?
Yeah, so this simulation, I think they're just taking the response level of the sort of the camera photoreceptor and using basically kind of a retinal simulation and creating outputs. And then those outputs are going to be used to drive the retinal ganglion cells. So in this case, this would work. So you notice you're getting really a sense of flow here. Right, so edges, static. Still can't read the Clorox can. You can actually, interesting, you can see sort of a bright reflection on the bar edge of the table. You still don't see the mass of that whiteboard very well because it's all one thing. Now you can probably find the door on the way out. And now you can see the rings. Did you read Clorox?
Yeah.
No, I think that's it. So really, but 64 by 64 is, you know, that's not a lot of resolution. You could imagine doing a lot better than that. So here's a video of a man who's received a visual plan. Right.
Yeah.
Oh.
Wow.
Can you really see?
Yes.
Oh my! So they just turned it on.
They're flashing.
They're flashing? Oh my goodness. That's so cool.
Larry Hester, 66, has been blind for half his life from a condition called retinitis pigmentosa. In September, an electronic stimulator was surgically implanted in his left eye. On October 1st, Duke eye surgeon Dr. Paul Hahn turned it on for the first time.
So the journey is just beginning. This is day one of hopefully the rest of his life. I think turning this device on allows him to experience a whole new world. It's not vision as we traditionally know it, but it's a whole new type of vision for him.
Are they ugly? Is that what they are? Only the coolest come on.
It was incredible. It was bright and it was significant. And I just had to take a deep breath, and I was just, it was hard to articulate what I was feeling, but I wanted to share it with everybody I could grab at the time.
Thank you for this opportunity for Larry to see a little bit again.
These patients have been essentially living in blackness in most cases for many, many years. And so what this is doing is almost, if you could imagine, almost a pixelated type of vision where they are getting stimulated with flashes of light when they perceive something that's bright, and when their camera is picking up something that is darker, then they get less flashes of light.
Yeah. Oh, look, you did it. You went right to it. Woohoo! All right.
That's pretty cool.
Well done,
I mean,
Larry.
that's significant. Wow, that's cool. That's great. Well, that's amazing.
She's more excited.
So,
I know, right?
what the flashes of light mean, and in many cases it can help them determine where a window is, light coming in through the window, or where a doorway is, and essentially help them navigate the environment.
It's so basic. The light is so basic and probably wouldn't have significance to anybody else, but to me it's meaning I can see light and we can go from here.
This is a milestone to me on many fronts.
You didn't see, I think there's the camera.
Individual level
Right
for
in the
Mr.
middle of
Hester,
the
it's a
glasses.
milestone because again, for the first time, he can experience something that hasn't experienced in 30 years.
And it looks like they've only implanted
I
sort
think
of one
this
side.
represents a new generation of medicine where rather than just treating or just watching as patients lose vision or trying to stabilize vision. For the first time, we can artificially restore vision. And I think this opens the door to a whole new generation or era of medicine, which almost makes sense to me.
All right, so one of the keys there was that in order to stimulate the ganglion cells appropriately, they actually had to know something about how the light is being processed from retina to bipolar to the ganglion cells, just like we talked about. All right, so I just wanted to bring up one more thing. Thus concludes Unit 4, but I just wanted to go back, since I know Unit 3 was like a long time ago, so I just was going to post a few questions, sort of guidance, since there were a lot of questions of, you know, what do I need to know from Unit 3, so back to synaptic transmission. So, you know, my recommendation is to really start with, you know, what were the fundamental concepts involved? What were some examples of those concepts? How do we know, we talked a little bit about experimental evidence for those concepts, and then sort of basic examples of pathways for each type. And so I have sort of a list of questions to kind of help guide you a little bit in those areas. So you can look over those.